correctly. Recording is now in progress. For those of you who are interested in sharing this later on, I will be posting the link. It probably won't happen tomorrow. It most likely will happen on Monday, but I'll post it to our blog and also on social media so that people can find it. So remember that your video is off, your audio is off right now, you're all muted. So you can use the chat to make comments. You can use the Q&A to ask questions. There will be no audio visual record of you unless at the end you ask to be unmuted to verbally ask a question. So you're all being recorded, but you're not really being recorded unless you do something later on, okay? And we've got both the chat and the Q&A. Remember that in the Q&A, if we have a question there and you like it, you can vote for it basically. And if there's a question that you also have, and it just helps us kind of keep track of which are the really popular questions to kind of make sure we definitely hit those. But you can also use the chat during this, and I'll be monitoring those and kind of passing things along as we go. So without further ado, to give you a little bit of background on Team Cooke NoMADS, this is a group of UNH Manchester students who approached us about doing some outreach on site. And so for several months now, they've been coming on a Saturday every month, setting up a table or doing an activity for our visitors. And they've also worked with school kids after school as well, which is wonderful. And it occurred to me that not everybody gets a chance right now to come and visit us, and that the work that they're doing in their project, which is pretty amazing, might be of broader interest. So I asked if they would be willing to do this, and they said yes, which is fantastic. So I'm going to turn it over to them in just a minute, but they'll be providing you with some insight, not only into kind of the science, but also the process in terms of working with NASA and doing what they do, which is pretty cool. So I'm going to turn it over to you guys now. I'm going to stop my video and mute myself.
All right. Thank you, Mirka, for that introduction and for having us here tonight. We're really excited to share about our project and our experience. So I hope you can all hear me and the screen is good. So my name is Sydney, and my co-lead Raymond is the other presenter tonight, the other little video that you can see. So we co-lead this team that is now up to 9 students, and then we have our faculty advisor, Dr. Cooke. And we just kind of put our team members' names here so you could see that we all have different positions and different backgrounds, which help us in different ways on this project. So the reason that we're here is because NASA put out a competition through their STEM on Station program called the SPOX competition, which was Student Payload Opportunity with Citizen Science. So they really wanted to get college students and other students interested in space science. So they created this competition and said they would send 5 payloads from college students to the International Space Station. So our faculty advisor, Dr. Cooke, read about this in a newsletter, and she thought it would be a great grant writing experience for Raymond and I. She didn't think we'd really get it, but we spent a lot of time, we worked on it. We developed the idea for like about a year because it got delayed because of the pandemic. And we finally submitted our proposal, which ended up being 28 pages, which was intense. And then we ended up being selected as finalists. So 10 finalists presented to NASA, and we got to meet with, it was a large board of people from NASA, which was really cool, a really good experience to present to them. And then we ended up being one of the 5 teams in the country that were selected. So this was the little announcement that they put on their Tumblr page when we got selected. And this was our original team. We had just 4 students. We've now added 5 more. So that's kind of the little bit of background about why we're here. So this is our current team, and we kind of have members coming and going as people graduate. This project is a lot longer scale than I think we anticipated in the beginning. So in the middle, we have Dr. Cooke, and then Ray and I on either side of her, and then our other team members. And the 2 little pictures on the sides are our newest team members who weren't part of the team when this photo was taken. And then we've got also a team member in the middle there that's no longer part of the team, but, and then we just had one join like Friday who is not in this photo at all. But our team is pretty dynamic, but also very dedicated. Those of us who are on the team are very dedicated, and I'm very appreciative to work with such hardworking students as well. So I will pass it. Oh, nope, sorry, this is my slide. Project overview. Basically, our project has 2 goals, 2 missions. The first is to observe the effects of the space environment on bacteria on the International Space Station. And we'll talk more about all of those aspects later. Basically, our hypothesis, though, is that the environmental conditions of space in combination with an in situ-like growth chamber will produce a higher rate of genetic mutations and modifications that will result in an increase in bacterial isolates that produce antibiotics as compared to an Earth-based control. And every time I have to say that out loud, it sounds horrible and mouthy, but that's basically our hypothesis. And Ray will explain all of that when we get to the science part. Our other large mission is to reach as many people as possible, specifically in New Hampshire, just because we're here, but also all over the place, and teach them about bacteria, antibiotics, antibiotic resistance, and space science to get people interested in STEM. So these are a couple of pictures from our early outreach programs, and we'll have plenty more as we go on too. All right, now I will hand it over to Ray.
All right. So like Sydney said, our hypothesis really is, well, our questions are to look at what are the differences in genetic mutations between Earth and space, and what are the differences in antibiotic production. So to answer those, we kind of need to review some scientific overview questions. So bacterial resistance is really what we're getting at. And the resistance to antibiotics is a huge and growing crisis. So the WHO has said that by 2050, approximately 10 million people per year will die because of resistance to antibiotics. This is a huge issue that will affect anyone from getting a tiny cut on their hand to people that have received transplant surgeries or chemotherapy patients. It's a much broader issue that we're really only starting to see the effects of. But how does this translate to space? So off Earth environments, we know that they have mutagenic effects. So an example of this is when astronauts come back from space, we can often see that they have, maybe some bone disorders where they have like weaker bones, right? So how can we predict how bacteria will change in space? So we know that larger organisms like humans change, but how do we know how smaller organisms change? And this is really important for long-term missions for astronauts' health. So say if we want to go to Mars or 6-month missions or 2-year missions, right? How do we know how bacteria will change? So it's important to develop this model of bacterial mutagenicity. But where are we going to get these bacteria from? Well, our team decided soil because a lot of our previous research with Dr. Cooke's lab is with soil bacteria. And number one, it's everywhere. And number two, it's free. So it has an incredible diversity of bacteria. There's so much bacteria in soil. And it's also a great source of antibiotic producers, which we'll talk about later. But another great part about our experiment is something called an iChip. So it was developed by Dr. Slava Epstein at Northeastern University. And basically, it's an in situ-like device that can culture more bacteria than normal lab techniques. So in situ meaning it's closer to like what a bacteria would experience in its own home environment. So let's talk about some general background information about bacteria. Bacteria are living organisms that are really small. They range from 1 to 10 micrometers. That's really, really small. So in comparison, the human hair is about 70 micrometers wide. That's tiny. And they're found in almost any environment. So both man-made and natural environments. Soil is a great source. It's a huge natural environment, and there's a lot of bacteria in it. But there's also a lot of different types of bacteria and shapes of bacteria. So you can kind of see some of them here on this page. They kind of look like rods or little squiggles or little balls that are kind of connected in chains or just lone bacteria. But there is a problem. So there's something called the Great Plate Count Anomaly. And basically what this is that we're not very good at growing bacteria in our labs. They like to just live where they normally live. They're really good at it. We're only able to really culture about 0.1 to 1% of all bacteria. That's a really, really small number. And realistically, most bacteria are harmless or beneficial. So let's think about our human microbiome. That's a great example. So all the bacteria that live on the outside of us, but also on the inside of us. And they help with things like our immune system on the outside and inside, but also basic things like when you eat something, you need the bacteria inside of you to be able to process that food right. So, all these commercials about probiotics, well, that it's very important. Your bacterial health inside of you is very important. So we need to be able to study that for astronauts as well. And less than 1% of all the bacteria that we've been able to culture and that we know about, which is already a really small number, cause disease. So it's an even smaller number. So, we also in our endeavor want to break that stigma against, all bacteria are really bad, right? They're really, really good. They really are. They're our friends. So when we talk about antibiotics, we're really talking about bacterial competition. So like any other species, bacteria compete for resources when they coexist. Soil is a great example of this. There's a lot of bacteria in there and they compete. They fight tooth and nail for all of the nutrients in there to be able to reproduce and to grow more. But to also kind of make sure that the other bacteria don't grow as well so that they have the space to grow. So they create these bioactive molecules, this antibiotics, to survive by selective pressure. But there's two sides to this coin. So while they both produce this antibiotic substance, they can create and activate genes that suppress that antibiotic ability. So they have these antibiotic resistance genes. So that's something we're also trying to look at. And interestingly, Streptomyces is a species that's very, very common in soil. And 70 to 80% of all antibiotics that we have ever created, you'd normally think they're created in a lab by a chemist. They come from Streptomyces in soil. It's really cool how we can use this natural product that's found everywhere, and we can then extract these antibiotics and use them for our purposes. And we can visualize these in a lab when we take soil samples and try and culture them by these things called a zone of inhibition. So you can kind of see in this picture of a petri dish on the right, there's little blobs in our grid sections. So like right under the letter B, there's a central little blob of like yellowish color. That's a soil isolate bacteria. And then around it, there's a little clearing and then more bacteria around it. That little clearing area is where the central bacteria has expelled and secreted or excreted, antibiotics or bactericidal agents. And there's that zone where nothing will grow. And that's how we can see that one is actually producing antibiotics. So why are we sending it to space? Well, space matters. It really does. And we know that space can affect single and multicellular organisms like humans. But how does it affect us? So there are things like electromagnetic radiation and microgravity that can cause selective pressure or spontaneous mutations. So bacteria is really good at growing where bacteria lives. And when you put it in an environment where it doesn't have all the things it needs, it kind of freaks out. It will try and figure out a way to survive in that situation. And this is where we can get our antibiotic production out of, is when it kind of gets stressed out. So we already actually know that the ISS is a great place to see bacterial mutations. There was a study done, I think it was published in 2021, where they looked at ISS services that were collected during 2015 and 2016. And they swabbed them and grew cultures and analyzed for microorganisms and bacterial growth. And what they ended up finding was this brand new bacteria. But, of course, it didn't come from space. That would have been an alien. You know, that would have been cool, but we know that those aren't that easy to find. So we know that it was a bacteria that came from Earth, went up to the ISS, existed there for a while, and mutated into something brand new. So we have this Methylobacterium ajmalii that was brand new and discovered just last year. Feels like this year, but just last year. And you can read more about it if you want to scan the QR code or copy the DOI number there as well. So let's talk about our payload design a little bit. And we'll also talk about kind of how we work with NanoRacks and NASA to get our mission kind of cleared. So this device on the left here, this thin little board looking thing, that's our iChip. So this was developed by Dr. Slava Epstein at Northeastern University. And it's a Teflon board that can be sterilized with little holes drilled in it. And these holes are where we can actually put growth media in, and then we can seed that growth media with our bacteria that we want to look at. And then we cover it with membrane material that only lets nutrients and water and oxygen through. So other bacteria can't get in those holes when we seal it off. And then we can put this device in soil. And soil is really wet and it's really good at holding nutrients in it. But it can also, through that moisture, carry nutrients into these little growth chambers through that membrane. And that's how we're creating this in situ-like environment. So instead of just trying to grow it in normal soil, we're able to isolate in a sterile environment bacteria and draw in the natural and environmental nutrients. It's a really cool design. But then on the right here, we have our design constraints given to us by NASA. So when they announced the competition, they gave us a real challenge. We had to design an entire experiment that would be out of our hands for over a month, that was in a 10 by 10 by 15 centimeter box. That's really, really small and really, really hard, especially for scientists that are used to a laboratory that's a huge room and all the room that we could ever want. And we have to make sure that it's automated. So when it goes up to the space station, we're not going to have any astronauts that interact with it. It's going to come off of the shuttle that brings it up there. They're going to install it into the ISS, and that's it. It's hands off until we get it back. So it's pretty incredible. But through this entire time, you know, it's been over a year since we've been working with this company called NanoRacks, and they help us get through the safety clearances with NASA. And, you know, that process has been really rewarding. It's been really cool to learn how science projects get from concept and idea to realization and to, you know, their launch date. And we're really excited for this spring when we will launch. I can't wait. I think most of our team is just really excited to be down in Florida and watch that spacecraft go up. But all right, let's talk a little bit more about payload design. So, of course, we're sending it to the ISS, but we need to make sure that it's safe. Like we mentioned earlier, we're sending up soil bacteria, but we actually won't know what bacteria we're sending up. All we're really doing is extracting bacteria from the soil and implanting it into this iChip. We won't know what they are until we get it back. So it's important that these astronauts are safe, because not only do they work on the ISS, but they live there too. It's their home. And you know, you wouldn't want some pathogen coming into your home and risking your health. So one of our constraints from NASA was a 3-level enclosure design to make sure that there's no chance that any bacteria can get out of this. So the diagram that you can see, it's a cross section, and there are these clamshell pieces that interpose on each other, and the joints around them, changed from side to side to make sure that nothing can get out of this box. And it's a really cool box, honestly. So we have a member of our team that just specializes on this 3D design. And we actually 3D print our enclosures out of a material called polypropylene. And it's a really cool material because it can be extruded and 3D printed. But then once it's assembled, we can also sterilize it. We can heat it up to a really high temperature. We can get it wet. We can clean it with organic solvents and also water. It's really, really resistant, and it's exactly what we needed. And we can design it to be exactly what we needed. So when designing, we knew that we wanted it to be extremely strong because it's got to go through the forces of a shuttle launch and also a return when it falls from space. So we designed it with something called a gyroid structure. And this is a mathematical model that we worked with one of our professors at the university to develop that maximizes the strength while reducing to a minimum the weight of this box. So some of the other pieces in this design, you can kind of see in the very center, there's a very small kind of button. Sydney, I don't know if you can hover your mouse over that. Yep, so right there. That is where we'll have a little data logger. So that's where we're going to track things like temperature and humidity. And it'll be sealed off and sterile from the rest of the environment. And then just right under there, you can see this thin little board with the holes. That's our iChip design. And it's nice and safely secured right there. And it'll be held in place for the entire duration of the experiment. And then under that, in that orange area that's open, that's where we'll put our soil. And of course, you know, it's unsterilized soil. It's one of the very first times that unsterilized soil is going up to the ISS. So we want to make sure it's right there in the center and nice and safe from the astronauts, and the astronauts are safe from it. So now we can go into some of our timeline and kind of our data that we plan to get from our experiment. So our experimental is what we know as what's going to go up to space. And then our control is what we'll have on Earth to compare it to. So different things that we'll measure. When it's on the ISS, we're going to take temperature recordings with that data logger. And that's going to enable us, when it gets back, to run our control so we can mimic it. Lessens the variables that we have to deal with so that we know we're getting consistent results. So things we'll measure are the number of isolates that produce antibiotic metabolites. So the way we'll do this is by getting our bacteria out of the iChip, extracting them out, and then culturing them in our lab and growing those zones of inhibition, just like we saw earlier. And that will tell us a number out of 384 little samples, how many produce antibiotics. And then we'll look at genetic identities. So we're actually going to sequence and extract DNA from all of these different bacteria. And there's two different sections of DNA that we'll look at. We're going to look at something called 16S ribosomal rRNA. It's a section of the bacterial DNA that's pretty much common among all bacteria. And we can cross-reference this section of DNA to known databases. There's a government-run and worldwide databases that compile lots and lots and lots of information from other scientists of known bacteria. And so when someone finds a new bacteria or they sequence a bacteria and find its DNA, they put it in this database, and then we can use that to see, okay, do we have a bacteria that matches one of those? Do we know if it already exists? And then we're also going to do this thing called a full genome sequence. So this looks at from A to Z, what is every single piece of this bacteria's DNA? And that's really cool because from that information, we can actually kind of work backwards and figure out, okay, well, it has this segment of DNA. It can make this portion of messenger RNA, which then can tell us what type of proteins it makes, which then can tell us what type of antibiotics it can produce. It's kind of cool how we can work from antibiotics to DNA, but then DNA to antibiotics as well. So we're gonna also do the same similar thing with our control group on Earth. We're gonna look at the number of isolates that produce antibiotic metabolites. And who knows, maybe there'll be a difference. That's what we're hoping. But maybe there won't be, that's data too. And then we're also gonna look at the genetic identities. So something that's really cool is we're not just gonna send up soil bacteria, we're gonna send up a couple known isolates. So we're gonna send up very specific bacteria that we actually already know everything about them. Well, pretty much everything about them. So we'll be able to know when it comes back, is there any tiny, tiny, tiny difference? And that difference will be because it was in space, which is really cool. So kind of going stepwise through our process. At the very beginning, we have our payload enclosure and iChip testing. That's kind of where we're at right now. Still working out some of the science and getting good foundational knowledge about how to really excellently execute this project. And then we'll get to building the actual enclosure. And then we'll have the opportunity to go down to NASA in Florida and build our entire project there right before the launch, which I can't wait for, to work in the NASA labs. That's going to be pretty cool. But then we assemble our whole experiment, and we'll hand it over to NanoRacks, who facilitates it being handed over to NASA and loading it onto the spacecraft that will go up. And then the launch day. And we're praying for first go around, but there might be some scrubs. We might have to wait a couple days or a week, but hopefully first time we'll be lucky. Which of course I'm jinxing myself right now, but and then about a month later, a little bit over a month, we'll get it back. And for me, that's probably going to be the scariest time, when it's just out of my hands. I don't do well with that. But then we'll get the payload back and it will be sent to UNH Manchester. And that's, I think, where the fun begins. We'll get it back in the lab and we can start our control experiment based on all the temperature data that we'll get off of the ISS. But at the same time, we're going to start unloading our iChip and start looking at the different bacteria that are growing in our growth media. And that's when we'll look at all these different, antibiotic production and DNA sequencing. And then we start analyzing our data. That's going to be challenging, but we have to get our reports out, and then we submit our final reports to NASA. A couple months later, and then from there, that's also another step. Is we have a lot of different products that we can experiment with for many months to years to come. That's what's really exciting about this project is we have a couple fundamental questions, but we can answer a lot of different things with the stuff we're sending up. Awesome. But first, it begins with our soil. Right? The soil is the most important part. So I'll hand it off to Sydney now.
All right. So because the soil is the basis for our experiment and the most important part, we didn't want to just choose any random soil, and we didn't want to make it not important and not as big an experience as we could. So we actually held a competition kind of parallel to the one that NASA held of Spock's. And we called it our Junior Scientist Competition. So we put out a call for proposals to all the middle schools in New Hampshire earlier this year, or earlier last year, I don't know what year it is, earlier in the school year. And we got proposals from middle schools in New Hampshire with middle school teams that had decided where they wanted to collect the soil that would go to space. And the winning team is pictured here. They are the decomposers. So they thought of that name and they wrote their whole proposal themselves, which their teacher is super proud of. And they chose to get soil from the Newington Town Forest, which just happens to be the oldest town forest in the United States. So we've got historical relevancy as well as hopefully new groundbreaking science, literally, from this experiment. And then I wanted to share a couple of quotes from their proposal because they were really showed their passion for science. So they wrote that we are very determined because when we went to get our soil, we did it in the rain, wind, and 48 degrees. It was worth it. And then you can also see just that sentiment even more echoed by the crutches of the kid in the front here. He tore a ligament and still crutched his way a quarter mile into the woods with us to collect the soil on this day. So they are definitely very dedicated and very passionate. They're not going to let anything stop them from getting this soil for us and getting it to space. And then they ended their proposal with, our tiny class is finally getting into something big that could change something even bigger. Good luck on your mission. So we just feel so honored to be able to present them with this opportunity to send soil to space and to be able to work with these this group of excited and bright young scientists. So we meet with them about twice a month. We have taught them about bacteria so far. We plan to teach them about antibiotics, antibiotic resistance, and all of our other topics of our experiment. We kind of take, we're giving them a very in-depth look at each of the aspects of our project so that they're very familiar by the time our experiment goes to space. And then when it does go to space, they will have a control set up in their classroom. So their control will be concurrent with the one in space, and ours will be after it comes back. When their control is done at the end, they will get to visit UNH Manchester and come to our lab. So they'll get to process some of their control samples in our microbiology laboratory, which we're really excited about for them. And then we'll have to wrap it up at the end of the year. But you can see from these pictures here, we've met with them 3 times so far, and we had them fill out a contract, and we filled out a contract of our agreements for working with each other because we wanted to frame it as more of a partnership. So we're on equal terms with them. Then we have swabbed some common surfaces around their classroom and around their school. We collected some soil for some preliminary testing, and then we found time for some dodgeball, and they beat us really, really badly. So in addition to the junior scientists, which is our more in-depth experience with a fewer kids, we have the NoMADS tribe, and that's our broad reaching program. So far we have run after school programs. We've run some at the McAuliffe-Shepard Center. We've run them at the Sea Science Center, at Girls Inc., at some libraries. So these programs they give students an opportunity to interact with science outside of the strict academic setting. So we're hoping that it just gets them more excited about science outside of being assigned things in school. So we get to talk to them about our bacteria, about antibiotics, about our experiment going to space. And then after we do a quick presentation, we give them a hands-on engineering challenge where they design their own payload enclosure. We also do school visits, which I try to align with the Common Core and the next generation science standards that the schools have to follow for their curriculum. So it actually helps supplement their curriculum for them. And rather than being detracting from the learning, it aligns with what they're trying to actually accomplish in their school year. And we've done some tabling events like Mirka was talking about at the beginning. So we've done a bunch at McAuliffe-Shepard Center and some other places as well. So those have been really great for interacting with the public and general ages, kids as well as adults. So far we've interacted with 349 people plus whoever's on tonight. But we aren't done yet. Coming up, we have more school visits. We just got 2 new youth educators on from our school. So we're able to schedule more of these visits now. We have more after school programs at McAuliffe-Shepard and at UNH Manchester, as well as other places. And we're actually gonna have a table set up tomorrow at McAuliffe-Shepard. So if you happen to be going to the Space Center tomorrow, you can see us there. In addition, we'll continue to work with the decomposers with all of the things that I talked about earlier. And we're really excited about all of these opportunities. Like I said before, we really just wanna reach as many people as possible. So if you are interested in keeping in touch with our experiment, figuring out what happens when it goes to space and when it comes back, you can go to our website. We have this QR code here. This has a lot of information about all the topics that we talked about tonight, as well as information about our past and future programs that you can sign up for. And it also has little bios of all our team members if you're interested in that. We also have social media accounts on Facebook, Instagram, and TikTok, as well as a Twitter account. So I'll just leave this up right now so you guys can get that QR code if you want. And at this time, we are ready for questions.
Yes, please. I'd love to answer any questions we have.
Okay, and so I want to just remind everybody that we've got the chat. You can put comments and questions in the chat, or we've got the Q&A. So we already have one in the Q&A, and the question is, in the design of your box, how are you ensuring both that the bacteria stay in, but that the conditions of space are not excluded? E.g., it gets exposed to radiation and other things.
Yeah, Sydney, do you mind if I answer that?
Go for it.
Great. So there's actually a few different things that we could answer this question with. So first and foremost, the way we keep our bacteria in is with our 3 levels of enclosure. But before that, on our eye chip, we have a very thin membrane that only allows that nutrient in and out. And that ensures that there's no bacteria that can get in from the outside environment, but there's also no bacteria that can get out. So the question went away, but kind of talking about the rest of that question where, how do we ensure that the enclosure doesn't limit radiation and, effects of space? Well, our project is really exploratory. And part of the thing about this experiment is it's done inside the ISS. So NASA has different programs that run experiments within the ISS and on the exterior of the ISS. And, we have to remember that the astronauts also live in the ISS. So there can't be an extreme amount of radiation, but there is enough that it does affect things slightly. That's one of the reasons that astronauts don't stay in space for really long time periods right now. But we're hoping to see some effect. And whatever that effect is, we can say, well, we have some more questions. Let's send another experiment up. This time, let's put it on the outside of the ISS. Let's boost that electromagnetic radiation even more. So now we have more of that electromagnetic radiation while keeping the same amount of microgravity. So increasing that one variable. So we're able to actually look at, what are the effects of electromagnetic radiation with and without. And the actual design of the payload with this 3D printed polypropylene. Polypropylene is really good at being passable. So EM radiation goes through those barriers very, very easily.
Cool. And so we've got a couple of things now. How did you come up with the idea for this particular experiment? That's a very big question, I know.
It is a big question. So I guess I'll start. We were first just really excited to have this opportunity, and we actually thought of a different experiment first. But the more we thought about that idea, the more complicated we realized it was going to be. And we were talking about programming chips and having mechanisms that dispense liquid, and it just was far too complicated. So our faculty advisor, Dr. Cooke, had heard about these devices, iChips, and we were thinking about using them in a class that we teach called antibiotic discovery. And then we just decided that, the size was convenient with the size of the box that we had. And if it's already in situ, why not send it to space, So it just seemed like a really straightforward, relatively simple experiment that would get us a really good chance of getting selected, and it ended up working.
Cool. And I would actually love to tag on to that question, because you mentioned how it was like a year of figuring this out because of the pandemic. So obviously that was one change, but were there other moments where you had to say, wait, this isn't going to work, or wait, we have a totally different idea, during the design process?
Yeah, definitely. I don't know, Ray, can you think of specific examples of that?
I think the biggest example is working through the problem of, well, now we have these bacteria in an in situ-like environment, right? Well, we talked about how are we going to grow them in the lab, because it's really challenging to grow them in a laboratory. So that's one of the things that we also had to work through. And, we've come up with some pretty ingenious ideas that I think people are just going to have to wait and see the answers.
Nice. Okay, so some other questions. Will you be testing for viruses?
We will not. So I don't know the full context of this question, but basically the media that we're using inside the iChip is specific to bacteria, basically. So viruses don't live without a host. So I guess if there were bacteria viruses within the soil, we could be also growing those in the iChip, but the way that we process on the back end has to do with culturing bacteria specifically. So if there were viruses, I don't think we know about them.
Gotcha.
I'm gonna kind of answer that question a little bit too.
Okay.
So along with what Sydney said, you know, we have that isolated bacteria in our iChip, but one of the great things about our experiment is we're sending up non-sterile soil. So every single thing that's living in that soil, fungi, viruses, every type of microorganism, we'll get back on from the flight. And we'll, you know, it might take us more time. It's not our initial question, but that's something for other students maybe that come after us to be able to use and experiment with. So I don't know, maybe they will be able to look at viruses.
That would be, I mean, I imagine there'll be all sorts of stuff that will be like, well, we didn't know that was going to happen, or we weren't looking for that, but it happened anyway. Yes. So now we've got it, right?
Yeah, definitely.
So do you think that the gravitational difference between Earth and the ISS will have an effect?
Well, you know, that's a great question. And I think that's, you know, our chief question. We really don't know. I mean, we have ideas. We can say we think it does, but that's what our experiment hopes to prove scientifically and empirically is, you know, with numbers, how can we quantify that effect?
Yeah.
Right.
I mean, and we did find studies at the beginning of preparing for this that stated that bacteria grow differently in microgravity, either simulated or real in space. So we know that they grow differently. So we're hoping it will have an effect, antibiotic specific, but that is actually probably the bigger piece of it. There's more, well, there's less gravity, but gravity will probably have a bigger impact than the radiation even in space.
Gotcha. So it comes back, the payload comes back, and now you finally get to see what's there and start working with it. How long will the analysis bit part take? I mean, is this the kind of, like, we're talking another year? Are we talking a month? What are you hoping for in terms of figuring out what happened? Do you have an answer? Maybe not. I
mean, we do have, I think it's a 90-day limit from NASA of when we have to return our final report of what our question was. But I think this could be a project that continues for months, if not years, once we get the preliminary data, which we have to process as quickly as possible so the bacteria don't revert. We can continue to do different experiments with that soil that went to space and all the different aspects that were there.
Gotcha. And so then piggybacking on that was a question that's already been liked, which is, would you come back and do a presentation on the results for the Discovery Center? And I second that one, or third that one.
Absolutely.
As well.
Yes,
we would
definitely.
love that.
That would be great.
That would be fantastic to kind of see and hear about lessons learned as well in terms of things. So I guess to follow up on the gravity, what is the difference between the Earth's gravity and the gravity on the ISS? Is it a big difference? I should know this, right? I mean, I work for the Discovery Center.
I can answer that because I included it as part of my kid presentations. So the gravity is actually about 90% of what it is on Earth, but because of the way that the ISS orbits the Earth, it's as if it's in a free fall around the Earth all of the time. So that's how it's experiencing microgravity.
Gotcha.
I hope that answers the question.
Yeah. I mean, I know that they talk about the astronauts up there. They're just constantly falling. It's just, but
that's what it is.
Yeah, exactly. So, so this is a comment. Did I hear that you are sending a bunny into space? I don't think
no bunnies.
I heard that part. No bunnies.
No bunnies.
Nope.
A bunny would not fit in the very small
that's a very
box.
small space.
Very
Absolutely
small.
Right.
Yes.
There we go. So if you got the chance, would you send soil to Mars to record changes? That's a very interesting question.
Yes.
So I don't know if we'd send soil to Mars, but we would love to send the technology that we're using to Mars to be able to capture some of, any possible microorganisms that might be on Mars. So that's one other thing that the person we work with at Northeastern University, Dr. Slava Epstein, is working on, is something that you don't have to put bacteria in, but that you put in an environment and the bacteria go to it. So this is, you know, this one question can lead to more experiments in the future, definitely.
Yeah. And so there are a couple of questions from the same person, and I'm going to do them in reverse order, but have you sent anything else up into space, you guys?
We have not. No, this will be our first.
It'll be exciting. What other experiments do you do in your lab?
Ooh.
A lot.
Yes.
Dr. Cooke is the idea person, and she likes to joke that every time we go to her office, we leave with more work because she just has so many ideas, which is definitely true. So I actually teach the lab of antibiotic discovery at UNH Manchester. So we definitely get a lot of research projects that base out of that. Mine in particular have been kind of smaller protocol type projects. So that's kind of some of the research. We have some people on the team doing research in microplastics as well. But I would let them talk about that. Ray, do you want to share about your experiment?
Sure. So I've been working on an experiment for, it seems like quite a while now, where I am actually looking at the ecology, so the diversity of bacteria from from a mountain in New Hampshire. So I'm actually more specifically looking, is there a difference in bacterial diversity at different elevations? Yep. And then, you know, once I answer that, I can answer different questions. Well, you know, are there differences in antibiotic production from different elevations on that mountain as well?
Neat.
Yeah. And then I also have something else to add. So in addition to like the mountains, last year our class went to the polar caves to sample to find cave bacteria that produce antibiotics. And this year we're going to a tidal creek in the Great Bay Estuary to find marine bacteria that may produce antibiotics. So we're kind of we're gonna get all over New Hampshire and then into space even. We're gonna find all the antibiotics.
Well, I mean, in some ways it's so, I don't know, I guess it's kind of heartening to me to hear about all of the places that you're looking, because we do hear a lot about antibiotic resistance. We do hear a lot about, oh, what happens when we don't have these options, that kind of thing. And so to hear like, well, we're looking, you know, we went into the caves that are down the road, or we're going into, you know, the idea that there are so many places to look for them feels kind of comforting to me in honesty when I kind of think about this. I mean, I recognize you've already talked about how difficult it is to grow them, you know, all of those things. But I'm like, okay, so there are many options here to explore.
Yeah, definitely.
So that feels very nice.
And actually, a couple of the coolest ones, I don't know if this will be comforting to people or not, actually, but some of the coolest bacteria you've seen come through our class came from a houseplant soil.
Yeah.
So
wow.
Yeah, it doesn't even have to be someplace obscure. Just something in your house could have the next penicillin.
Oh,
that's very cool.
To kind of add to that, so one of the things we recently did with our junior scientists is they touched a bunch of petri dishes with their hands, and lo and behold, those petri dishes were full of bacteria that were producing antibiotics. So just from their hands, we found antibiotic producers. They are everywhere.
Everywhere. Okay. I mean, that's kind of uncomfortable at the same time, but I'm good with it. I like it on the whole.
Yeah. Healthy bacteria on their hands.
Right, exactly.
Healthy, yes.
Since this is such a long-term project and you are students, will you continue to follow this after graduation or pass it on to other students? It's kind of a big question.
I don't know yet.
I think, to be honest, it's kind of the opposite. So my academic career has been more shaped by this project than it being shaped by my career. So I am in the master's program at UNH Manchester specifically to work on this project and to keep working with Dr. Cooke as well. So it's kind of like, I think I'm here until it's done, probably, even if I'm not a student. I don't think Dr. Cooke can get rid of me, but but we do have other people on the project that are moving kind of through, like we have people join and help, and then they'll graduate and move on to different things.
Yeah, I do think I've seen interviews with other groups working with NASA where they talk about the long-term, you know, time frame. And I think about those people who planned, you know, years ago for a launch to happen so that 7 years later, the thing that they were planning for could happen. And that whole waiting process, and, you know, you wouldn't want to walk away from the project, but you have to wait for so long. It's such a long process. I think it must be really difficult to kind of see, you know, how things progress and stick with it depending on the time frame. So.
Yeah, it's been a lot adjusting to the stress of such a long-term project as well. You know, I'm used to a semester and then I get a break, but this has been pretty much constant.
No breaks.
Okay, so we have some specific questions about the experiment. Is there only one iChip in the box?
So that's an interesting question. So the iChip is built with an array of 384 holes. So we actually have divided that up into 4 little sections of 96 holes per section. And on those, we call them quarter iChips. We can actually break up and implant them with different types of bacteria to answer different types of questions. So kind of like the same iChip, but there's only one contact surface with the soil, if that makes sense, if that answered your question.
Is the data logger an off-the-shelf commercial unit? If so, what is it?
It is.
It is.
Do you remember what it's called?
Oh, geez. You know what? I don't remember the exact name. Oh, it's actually it's called Track It.
And I'll tell you, on it.
It is it's a New Hampshire company that made it.
Oh, neat.
So there you go.
That's very neat. All right. I love that. Is there anything you are anticipating to find in particular from this experiment? You
know, we're hoping to find any data. Any data is the
Spoken like
thing.
A true scientist. We are hoping for something.
You know, I think one thing we're taught a lot as scientists is don't anticipate results, because then you're idealizing your project. You're trying to fabricate data where it may not exist. So we'll just have to wait and see what happens. Right. So hopefully, if everything goes well, something will happen. Something, yes.
Something, yes. Yep.
What is in the sections of the box above and below the iChip and soil? It looks like one data logger. Are you putting in 2 for redundancy?
That's a great question.
Let me go back to it. The diagram's a little bit confusing.
Yep. Yeah, we can walk through that diagram a little bit more, definitely. So
this is when animations are annoying.
I'll answer the second question. No, there will only be one data logger. We're very, very confident with its functions, though. We've tested it a lot. It does very well. It's extremely long battery life. We're going to be taking tens of thousands of data points to measure temperature, to get a very accurate sense of the actual temperature fluctuations on the ISS. And you'd be surprised, it actually fluctuates quite a lot. It can change anywhere from 5 to 15 degrees throughout the day. Fahrenheit, not Celsius. So
since I have the mouse control, I can explain the diagram here, which is a little easier. So this is the data logger. It's actually the one we're using is a little bit bigger, but this is an older diagram. So this will be a data logger. This space in this most inner line is going to be the gyroid, and the same on the bottom here. That will be filled with gyroid. And then you can see, like, if you follow this next section around to where these are the joints, this is the innermost level of containment. So that's all solid polypropylene. And then the next one, the next one up is also solid polypropylene, the next layer. And then there's a third around that. Did that help?
Yeah. Cool. Yeah, I think it helped to kind of go back through it now that I've heard more about how things are being organized and everything. Okay, what is a gyroid? Would be a follow-up question to.
I also answered this one for the kids, so I'm going to steal it if you don't mind, right?
Yeah, go for it.
So I have heard Dr. Plant say, and I don't ask me to explain this, but it is 2 interposed sine waves in 3 dimensions.
Yes.
So a lot of math. It's a lot of math, like hours of math that he does. But basically, like, the sine wave does this, and then imagine one going like over it, and then like all 360 degrees of sine waves interposing with one another. So it's basically super stable. It's able to 3D print really well because there's no support needed. If you're familiar with 3D printing, you can't print over nothing because the melted plastic would fall. So the angles work really well for 3D printing. It provides a lot of airspace and keeps it light while also being really strong.
Yep. And if you kind of look at it from one angle, it kind of looks like a saddle shape in some orientations, but kind of like oscillating saddle shapes, if that helps you visualize it a little bit.
Well, I'm sure we can also look it up, see if we can find some other images for it. How is this project being funded?
Yes, so that was part of the competition. They allocated, I think I could say $20,000 to each of the teams. So they decided, they decided to dedicate $100,000 to the whole thing. So there are 5 teams that are sending up projects. So we get $20,000 from them. And then we also buy things with our own money sometimes, like little marketing things. Dr. Cooke buys a lot of things, so we're grateful for that. And then we're also applying for other little grants that we find along the way. We actually just got approved for a grant for specifically the outreach materials, which is really exciting as well.
You would be surprised how quickly $20,000 can go in the science world.
I'm sure. Well, especially when you talk about kind of the process of figuring it out. It's not just that the 20,000 were for the final materials, but the 20,000 is for the whole process and the testing and the, you know, all of those pieces. Oh, how can we contribute is the next question.
Oh man. I don't know. Dr. Cooke, are we accepting donations?
If you would like to get in touch with us, please visit our website. And there's a questionnaire box that you can fill out and give us contact information if you'd like to know more or just to get in contact with us in general.
There you go.
We can answer that question later.
There we go. Does what you do involve coding?
So when we started this project, long time ago, it seems, we were going to do coding, but I have very little to no computer science background. Neither does most of us on the team. So we really tried to stray away from that side and tried to simplify it as much as we could.
So if I'm understanding correctly, then this will be launched, it will go up, it will simply be put in place, and then it just does its thing. There's and then comes back to you.
Yep.
Gotcha.
Yep. The bacteria are growing at room temperature on the ISS, and they're just doing their thing independently. And then we take them back.
You may not know this, but the question is, what model of rocket will it be going up in?
Yeah, we don't know yet. They keep that information pretty locked up tight until we get closer to the date. So we don't know.
Have they, I mean, not that you may not be able to say this, have they given you kind of a time frame for when the launch would be happening, like a range or anything?
Spring.
Okay. I mean, I feel like given other NASA launches, that's a reasonable range there. I mean, as I recall, you know, other things, there's a range, so that works out. Are any of you looking to work at NASA?
Well, one of our team members would love to someday be an astronaut. So yes. Yes.
She wants to study plant biology.
Cool.
Specifically.
Very cool.
The rest of us are just kind of existing. I don't we all have like different ideas. Some people like me have no idea what they want to do in the future, so maybe. But one of them definitely.
Cool. And so we've got a message from Dr. Cooke that says, if anyone wants to donate to support Team NoMADS research or outreach work, you can direct it to UNHM to the attention of Dr. Sue Cooke, that's me, and or Team NoMADS. I can tell you they will put it to good use. So there we go.
Thank you.
And I feel like, let's see, we got a couple more questions from the same person. So we'll kind of wrap up because they're wonderful questions. So what is your biggest goal this year is one of them.
Oh man.
That's a very big question.
Is it project specific or?
It just says, what is your biggest goal?
I think my biggest goal, I can answer that first in terms of this project at least, is to make the junior scientist experience as amazing as it possibly can be. Like I might want to go into STEM outreach in the future as well. And I will never have another hook like we're sending dirt to space. You know, I'll never have this opportunity to get people interested like I do now. And I'll never have the opportunity to work so closely with a classroom of middle schoolers and to provide them with this really cool opportunity. So I really just want to make things as amazing as possible for them and not think of anything after, like, oh, if only we'd done that. So that's my biggest goal.
Yeah, I think, you know, I kind of have a similar goal in that I want to make sure that I remember that everything we're doing right now, this is our time. This is it. We're probably only going to ever send something to space once, and to do it at all is incredible. So my biggest goal is to keep that in mind and use it to, you know, fuel my passion for both the science and the outreach work.
Nice. Okay. So we've got some other things. When later researching bacteria in Great Bay, might you be working with the Coastal Research Volunteers and/or New Hampshire Sea Grant?
I'm not sure. Dr. Cooke got us that connection. She used to research in the marine area, marine biology, when she was in her PhD program. So it's her connection. So I'm sorry, I can't really answer that one. Maybe she can comment if she's still here.
Are there any other space-related projects you're planning to take part in?
Not at this time. Maybe one day, but I think one is enough to tackle at
it's
one
pretty
time.
fantastic, right? Yeah. Okay, and then I saved this one for last because I love the idea of what we're ending with. Oh, and yes, definitely hoping to work with Sea Grant on both antibiotic discovery and microplastic work. Oh, very cool. So is this a fun project?
Yeah. I mean, at sometimes more than others. But overall, yes, it's very fun. I love just the hands-on working with bacteria stuff. I think that's way more fun than the like analyzing chemicals side. So I just like growing up little bacteria and looking at them. The working with the kids is the most fun. I can't believe how much fun it is every time I like spend time with kids after. I'm just so excited and happy. So definitely yes overall. And Ray, you can answer too.
Yeah, it's awesome. You know, of course, with anything, there's ups and downs, but you know, I think my favorite part is the problem solving that you have to do and the anticipation. So, you know, at some point, this is going to be out of our hands for 60 days, like really out of our hands.
Right.
And so it's, for me, the most fun part is to be able to figure out all the little details and find all the little things that might go wrong and how we can solve them before they go wrong. I think that's really fun.
Fantastic. Well, I think people have really enjoyed your presentation. And I was fascinated and I love hearing about the process, about figuring out one part, so then you have to do it this way and thinking through the problem. I want to thank you both for taking the time to do this presentation. I'm going to reiterate that I definitely want you to come back when you have done some of the analyses and let us know, you know, what
absolutely
what you found, if something happened. Hopefully something happened.
Oh, definitely.
Or maybe they'll all die and then we'll just tell you that.
It's very possible.
I know, right? Oh my goodness. But thank you so much. This was fantastic. I want to let everyone know that if you're the kind of person like me who thinks about questions 10 minutes after the meeting is over, you will all be getting an email tomorrow from me thanking you for being here in the audience and also asking for comments and suggestions and things like that. So if you have any questions for them, anything else you were curious about, you can send those to me and I'm happy to pass them along. Or you've got the website right there to contact them directly. And I want to thank everyone for joining us on a snowy winter night, and hopefully you have a wonderful weekend and stay warm out there. And we look forward to someday seeing you in person. It will be very nice, I think, and novel to actually be in the same place together. So thank you, everyone. Have a wonderful night.
Thank you.